Megazyme provides an extensive range of assay kits for use in various assay formats, including autoanalyzer, microplate and manual spectrophotometer. The D-Xylose test kit is a novel method for the specific, convenient and rapid measurement and analysis of D-Xylose in plant extracts, culture media, supernatants and other materials. This video tutorial will demonstrate the use of the kit with a prepared plant sample using the manual spectrophotometer format. The kit contains sufficient reagents for either 100 or 200 manual assays and is supplied with a detailed data booklet. The assay is specific for the measurement of D-Xylose. The principle of the enzymatic reaction involved in the measurement of D-Xylose is shown in this figure. The amount of NADH formed is stoichiometric with the amount of D-Xylose in the sample. It is NADH formation which is measured by an increase in absorbance at 340 nanometers. Prior to sample analysis, the kit components should be prepared as described in the kit data booklet, and once prepared they are ready for use in the manual assay procedure. All of the kit components except bottle 2 are used as supplied. The contents of bottle 2 are dissolved in 21 milliliters of distilled water. The bottle is capped and the contents are mixed thoroughly to ensure complete dissolution. For longer term stability, do not dissolve the contents of the second bottle until required. Follow the manual assay procedure as described in the data booklet. This demonstration is using 4 sample tubes, 1 blank tube and 1 standard tube. A blank reaction and a standard reaction must be performed with each batch of samples. Pipette 2 milliliters of distilled water into all assay tubes. Pipette 0.1 milliliters of distilled water into blank assay tube. Pipette 0.1 milliliters of sample to sample assay tubes. Pipette 0.1 milliliters of kit standard, bottle 5, into standard assay tube. Pipette 0.4 milliliters of bottle 1, buffer into all assay tubes. Pipette 0.4 milliliters of bottle 2, NAD plus and ATP into all assay tubes. Swirl to mix the contents of bottle 3 hexokinase prior to dispensing. Pipette 0.02 milliliters of bottle 3 into all assay tubes. When all of the components have been added, mix the tube contents thoroughly and incubate the tubes between 25 to 37 degrees Celsius for approximately 5 minutes. After 5 minutes, record the first absorbance reading, A1, at 340 nanometers for all of the assay tubes. In this demonstration, we are using the Megaquant-Wave spectrophotometer set to read at 340 nanometers. Alternatively, a recording spectrophotometer with a 1 cm path length can be used. Swirl to mix the contents of bottle 4, xylose dehydrogenase and xylose mutyrotase prior to dispensing. After recording the A1 absorbance value, pipette 0.05 milliliters of bottle 4 into all assay tubes. Mix the tubes thoroughly and incubate them between 25 to 37 degrees Celsius for 6 minutes to allow the reaction to go to completion. Record the absorbance reading A2 for all of the assay tubes. This absorbance reading completes the measurement of D-Xylose. The absorbance readings A1 and A2 of the sample and the blank reactions are used to calculate the D-Xylose concentrations in the original samples. When performing this test using the pre-installed protocol on the Megaquant wave spectrophotometer, the results will be automatically calculated and printed via the onboard printer, or the data can be exported to a computer using the SF Capture software. Please see our Megaquant wave video for further details. If the results output are raw absorbance values for both blank and samples, the calculations of D-Xylose content can be performed manually, as described in the calculations section of the kit booklet. Megazyme has also developed specific Excel-based MegaCalc applications for each Megazyme kit to allow quick and easy results analysis. Results can be analysed using the MegaCalc application specific to this D-Xylose kit, which is available to download free of charge from the Megazyme website. The MegaCalc spreadsheet provides full instructions for use. Open the MegaCalc worksheet and input the following: sample details, absorbance readings for the blanks. For each sample, input the sample identifier and the absorbance values for the samples. Alter the sample volume if a volume other than the default 0.1 milliliters is used. If dilution of the sample has been performed, then input the dilution factor used. If no further dilution was performed, the dilution factor is 1. When all of the data has been entered, concentration of D-Xylose in the sample is automatically calculated and given as grams per litre in the original sample. For solid samples, input the concentration of the original sample extract in grams per litre. The concentration of D-Xylose in the solid samples is then automatically calculated and provided as grams per 100 grams in the original sample. To calculate the arabinoxylene content in the sample, the percentage of D-Xylose content of the arabinoxylene must be known. Input the xylose percentage. Arabinoxylene is then automatically calculated as grams per 100 grams of original sample.